Okay, hello everyone. Welcome to the Great Lakes Seminar Series, co-hosted by the Cooperative Institute for Great Lakes Research, CIGLR, and the NOAA Great Lakes Environmental Research Laboratory. My name is Greg Dick and I'm the director of CIGLR. It's my sincere pleasure to introduce today's speaker, Dr. Cody Sheik. Dr. Sheik is an assistant professor in the Swenson College of Science and Engineering at the University of Minnesota, Duluth. He's a geomicrobiologist whose work in aquatic systems is at the intersection of three fields: microbiology, ecology, and geochemistry/geology. His lab currently investigates the microbiology of the deep subsurface, sediments, surface waters, and harmful algal blooms, which we'll hear about today. His research seeks to understand how microorganisms mediate biogeochemical cycles with an emphasis on carbon, nitrogen, and sulfur in these disparate ecosystems. On a personal note, Cody did a postdoc in my lab here at the University of Michigan, where he was a real leader of the lab, and I'm delighted to see him now doing well at a CIGLR partner institution. And finally, his promotion to associate professor with tenure was just recently approved, so congratulations, Cody. Before I turn it over to our speaker, I'd like to remind everyone to please leave your microphones muted for the duration of the webinar. You're welcome to enter questions into the chat box at any time during the presentation, and we'll moderate that at the end of the presentation. With that, I'll turn it over to our speaker. Welcome, Cody.
Great. Thank you very much, Greg. I really appreciate the invite. Yeah, it was really a great time. I was just thinking back, sunny days and Ann Arbor. This was quite the time of year to be there. So thank you all for joining us and joining me here. I'm excited to share some of the work that we've been doing here on Lake Superior, thinking about just the microbiology of the lake, which happens to coincide with the cyanobacteria story that's developing right now. And so today's talk, I'm really going to focus in on some of the work that we've been doing, thinking about cyanobacterial blooms in Lake Superior. And so that's sort of like the present part of the talk. And then we're going to switch gears and really start thinking about these blooms over time and dive into the sedimentary record a little bit. And then finally, we'll end up hopefully at the future part and really think about where the lake is going, hopefully. And so with that, let's get started. Today's talk is really going to be in like two parts. So the first part I'm really going to focus in on is the genome analysis of Dolichospermum limmermanii. And so this is the primary bloom forming organism in Lake Superior that has been here and reoccurring now for several years. And so we recovered a genome from the 2018 bloom, and I'd like to share a little bit of insight that we've gained from having this genome. And then Act 2 is really going to use that information that we pulled from the bloom and really start thinking about the broad distribution of this organism across Lake Superior and ultimately with time as well. So hopefully by the end of this, again, like I said, we'll get a better sense of the ecology of this organism in Lake Superior. So I'd like for you to like sort of picture a bloom. And you know, I think a lot of people here on this call probably think about cyanobacterial blooms in the Great Lakes and maybe think of like Erie as the poster child for this. These blooms in Erie, you know, they come off so routinely that you can kind of set your watch by them. But the lakes here in Lake Superior, these blooms are of a little bit different nature. They're quite ephemeral. And so these things come and go so fast that unless you're there with your sampling gear in place, you may never ever be able to get a sample of these things. And so I think this has been one of the hardest parts about trying to understand these blooms, is that we don't get the ability to go out and just routinely sample them. It's sort of a mystery still about why we see these things in different years, and the next year we won't, okay? And so kind of keep that in the back of your mind as we go through this, is that these things are of a bit different nature, I think, than what we normally would think of about a bloom. But that also kind of makes them fun, and you get to kind of be like Sherlock Holmes a little bit. And so my lab uses molecular tools, primarily DNA sequencing and RNA sequencing, really to understand microorganisms in the environment. And from that, we like to infer or get insights about the ecology of them, not only just like distributions across the landscape, potentially across time, but also to think about their function, right? What sort of metabolic genes do these organisms carry, and how that fits in the greater part of the ecosystem. And so with blooms in general, we know that they are increasing globally, both in intensity, duration, and frequency. And so this is a figure from Ho et al. A few years back. And the big take home here is if you look across their map, and these are focusing mainly on lakes that have microcystis blooms. But you can see there's a lot of red here relative to blue. And this is just showing that these lakes, the blooms in these lakes are intensifying, right? And what's interesting though is that when you start breaking it down by lake basis, and here I've just pulled out Lake Erie, Huron, and Michigan. These lakes are being driven, these blooms within these lakes are being driven by different environmental factors, right? So we see Erie had a long period of improvement. I think a lot of people on this call could give a laundry list of reasons why, but we also see this rebounding effect that's happening here. While Lake Huron has seen this sustained improvement, and Michigan has just kind of seen this flat line throughout these years. And I think the really interesting thing is that there's really no one driver here. You can't just say it's phosphorus, you can't just say it's nitrogen, you can't just say it's temperature within each one of these lakes because they're own separate entities, and the microbes and other eukaryotes are all responding differently within these. And so I think it makes it an interesting sort of study case to think about when we start thinking about Lake Superior. We know it has a large temperature effect, but we also know that it's also oligotrophic, and so the nutrient concentrations, primarily of phosphorus, are very low. So let's switch gears and start like really thinking about broadly about this growing problem of cyanobacteria in Lake Superior. And so starting in about 2012, there's been a real upsurgence, I guess. We started noticing blooms happening, I guess is what I should say. Don't want to get fancy with my words. But we've also seen the press really take hold of this, both at a local scale and also at a national scale. And I think this really feeds into the idea that we think about Lake Superior being this very pristine lake compared to, say, Michigan or Erie, primarily because we haven't seen big invasive mussel invasions or harmful algal blooms that are happening, shutting down water systems. You know, most people mindset when they think about Lake Superior is that it's large, it's cold. I can attest to that, swimming in summertime. But it's also like very clear, very clean lake, right? And because of that, tourists flock to this region during the summer to partake in its beauty, right? And so I think there's a lot of implications for these blooms in this region, both from an economic standpoint, but also from an ecological standpoint. And so it's really piqued the interest of researchers in the area of recent, and we're really trying to understand these things. And I don't know, there might be also a correlation between these cyanobacteria just really loving publicity. I think that might be also a correlation here. I don't know. That's just a hunch. But anyway, so our colleague, Brenda LaFrancois from the National Park Service, put this map together. And she's been really the boots on the ground for a very long time and has really brought these blooms to the forefront of everybody's thoughts in the region and also across the nation. And so she put this map together of both historical observations and some of the recent observations that we've had of cyanobacterial blooms. And here we have it color-coded by year, and this open circle here is just anecdotal historical observations. And so you can see we had some historical data showing that there has been blooms here on the north shore of Lake Superior. This is just generally the western basin. We have the central, and then we have the eastern basin over here. We've had blooms up in the bays of Thunder Bay and Black Bay, but you can see there's a high concentration of blooms happening here around the Apostle Islands. So this whole region right here is the Apostle Islands. And that starting with that 2012 bloom, it's sort of buried. It can just kind of barely see the tip of it right here. But you can see all these blooms are happening over different time periods. But the most extensive bloom that we've had so far has been this 2018 bloom. A lot of these blooms come and go very quickly, and so it's really hard to go out and sample them. But this 2018 bloom was pretty substantial that year, and it actually allowed us to go out on multiple days and capture samples from this bloom as it evolved over time. And so some of the first accounts of what it looked like here was this at Cornucopia Beach, and you can see it over here on the map. Cornucopia Beach is right here. The Apostle Islands are up here. Duluth is right down here at the very tip here of the lake. But you can see how green it is. And you can see actually people, like this bay is usually pretty clear. You can see the water, see all the sediments down here in the bottom quite easily. And so you see the number of people that are out here visiting this area, wanting to interact with the lake. And they are still interacting with the lake. We see somebody out here, maybe on a jet ski, I'm not really sure, but they're having a good time despite the fact that this water is, which is normally clear, is now green. There were also, by satellite image, you can see here there could be potential other blooms happening further down here at Wisconsin Point. But Bob Sterner's group, this is from his paper in 2022, they were actually, or sorry, excuse me, 2020, they were actually able to go out on day 2 of this bloom, and they actually sampled from Mawaki Bay, so right here, just well, a little tiny ways up from Siskiyou Bay. And they identified this organism as Dolichospermum limmermanii, and they were able to take some samples from within this bloom. And so this is the first real, like, accounting of this bloom and the organism that's here. On the 3rd day of this bloom, we were actually able to intercept this. And so my colleagues, Andy Bramberger and Chris Philstrup, they were actually measuring, I think, light in the water column aboard the Blue Heron, and they heard about the bloom and they hightailed it over. They had minimal sampling equipment, but they did find enough bladders that they could actually collect some water and bring it back to Duluth with them. And so then when they got there on the 3rd day of this bloom, it had started dispersing out with current and whatnot, but they were actually able to sample from 5 different locations, one here at Sand Island. They did a transect going into Siskiyou Bay here, so they did one way offshore and kind of as close as they could possibly get within Siskiyou Bay without beaching the Blue Heron. And finally, there was some accounts that there could be some other blooms happening out here on the other side of Bark Point, and so they took a sample over here by Herbster Beach. And so when they brought these back, well, sorry, get ahead of myself here. While they were out on the water, they were seeing these surface scums, which is pretty indicative of these past blooms of what people have seen. When they were doing some net tows, they found a very highly enriched upper water column. You can see how green it is there. And when we got it back to the lab, looking under the microscope, we could see the indicative chains here of Dolichospermum, and here's what an actual larger colony would look like here. But these images are very in line with Dolichospermum limmermanii. So it's showing us that our samples did have some, even though it was dispersing, we did still have some Dolichospermum hanging out. Okay, so when we got this back in the lab, again, my lab focuses on using DNA approaches to understand microorganisms. And so what we did is we took that water and we filtered it down onto a 0.22 micron filter. We extracted the DNA off this filter. We use some harsh chemicals and some jostling around, and we try to get as much DNA extracted out of these cells as possible. Because we're looking at a bulk sample, we're trying to get a snapshot of everything. So not only the Dolichospermum, but any other bacteria, eukaryotes, viruses, whatever's in that sample, we're trying to get that signature of the DNA out. We then sent this off to the sequencer here at the Twin Cities, University of Minnesota's campus, and we did a technique called shotgun sequencing. So here, what we're trying to do is sequence as much DNA as we possibly can at random, right? And we're using these very, very short DNA sequences that are 150 bases long. And to compare this to what a genome is, a typical genome is going to be somewhere on the order of like 3 million base pairs, if not even more. And so the big hurdle is to really take these little short sequences and try to overlap them in a sense to create a bigger piece of DNA. And so we use these algorithms to assemble this data back in together, right? And ultimately what we want to do is once we have these big sequences, we can start to associate them with other sequences that are coming from the same organism. Because it turns out DNA encoded within it is almost like a fingerprint. And so if you can read that fingerprint in just the right way, you can associate one piece of DNA with another piece of DNA coming from the same genome. And when we get these genomes, we call these things metagenome assembled genomes. Okay, and so once we have these genomes, we can do a lot of different styles of analyses. We can think about the evolutionary history of these organisms by building phylogenetic trees. We can use public databases and think about their global distribution. We can think about their metabolic function. And in the case of cyanobacteria, we can think about their secondary metabolite genes and toxin production genes. So is species A producing microcystin, or could it be producing things like saxitoxin or anatoxin? Okay. And so using this approach, we were able to assemble from these 5 different samples. And when we started looking at the cyanobacterial fraction of what we pulled out of this, it made my job very easy. I think this was probably like the easiest fishing search I've had to do for an organism. And so when we look at this, here on the left, we have 16S genes that we pulled out of the assemblies, and here we have genomes. It was remarkably easy because we only had 3 of each, so 3 16S genes and 3 genomes that were part of these assemblies. And so up here at the top, these top 2 here are typically what we think about as picocyanobacteria, and these are just naturally occurring genomes and organisms within Lake Superior. Currently, we have about 70 of these genomes from across Lake Superior that we've been looking at, and we keep adding more, it seems to be. So there's a lot of like this diversity of this group just naturally within the lake. But what caught my eye was this 3rd one here. And so by 16S taxonomy, we're using 2 different styles, I should say. We're using 2 different styles of databases. One is a 16S database, and this is a very extensive database. It's focused only on the 16S rRNA gene, which is a very conserved gene across all bacteria and archaea. And so we can use it as a really good taxonomic marker. And so we have the SILVA database, and we also have a genome database, which is this GTDB taxonomy. And so they have their own taxonomy that they use. And unfortunately, in some cases, the 2 don't overlap. I think there's a lot of people working on this, but it could always be improved, especially for the cyanobacteria. And so this really caught my eye was this Anabana cCAP sequence that was coming out, and we're identifying it across the board here at all of our sites that we sampled at. And the same thing with this LE011-02. If we looked at the taxonomy of this fell within the Nostoc ACA. And so I had a hunch that this might be our organism, but we really needed to build a phylogenomic tree to really see where this genome actually fell out taxonomically. And so I'm going to skip over the 16S genes, and we'll come back to those here in a bit. But let's just focus here on the genome and thinking about its overall taxonomic placement here. And so building this phylogenomic tree, we used around 60 single copy proteins from the entire Nostoc ACA genomes. So I think there was about, I can't remember, 170 or 200 genomes available. And so we just threw them all into a big tree just to see where the sequence would come out. And in this green clade is where our sequence is what we're calling SB001 is where it came out at. And everything here you see in green is all one species by average nucleotide identity. And so we can think about this whole clade up here just being every single genome within here would be potentially a substrain of Dolichospermum limermanii. And I should also note that we don't have actually a genome from a culture yet in any of the databases. And so it made it a little bit extra tricky, but I think we got there. Anyway, and so What was interesting, if we zoom in here, there's several things that kind of popped out to me as I started analyzing this clade. Again, each one of these genomes would be a subspecies. This upper clade here, where our organism, the SB001, falls out, is also closely related to this LE00011-02 and this AL09. And interestingly, this one is coming from Lake Ontario, and this one here is coming from Lake Erie. And so it seems like we have a nice Great Lakes, very similar genomes occurring in these three lakes. If I was a betting person, I would probably say that we'd also find these in both Lake Michigan and also Lake Huron, just as a hunch. We seem to find it everywhere. It's in Wisconsin lakes. It's also in Minnesota lakes. And so it does seem to be like one of these organisms that's sort of made its way across the upper part of North America so far. And what's interesting also within this clade, we have organisms or genomes / organisms on isolates that are coming from Finland and also the UK. And across the board, we see anabainopeptin is the primary potential toxin that these organisms harbor genes for. And so every single one of these genomes can produce anabainopeptin potentially, but only a handful of them can actually do microcystin. And so I have the microcystin in the star shape right here. And so this is actually quite in line with what we're seeing with microcystis, where you have some strains of microcystis can produce microcystin while some of them can't. And so I think this is a very interesting sort of dynamic. And so far, knock on wood, we have 4 genomes coming from North America, but as we get more, hopefully, most of these don't produce microcystin, would be the goal. But I do think it'll be interesting as we expand this clade out to see what some of these broader patterns of microcystin production actually hold here. And so one of the other things that really more broadly spoke to me about these organisms, these genomes, is that they're coming from very disparate environments just across the Great Lakes. So if you think about Lake Superior, this organism is apparently thriving here, but also potentially is thriving in Lake Erie. And so we know that Lake Erie has, for a long time, had Dolichospermum blooms in the Central Basin and sometimes up in the north, near the Detroit River. It shows up, and so we know that there's Dolichospermum there. We know there's Dolichospermum blooms there. Is it this organism? I don't know, maybe, but it could also be another one. But I do find it interesting that we have these two very in-member parts of the Great Lakes being high nutrients versus low nutrients. And so one of the big questions I had with looking at this organism and its genome is, how is this organism able to survive / thrive and bloom potentially in both Lakes Superior and Erie? And so we can think about Lake Superior is very low in phosphorus. We have much higher phosphorus concentrations in Lake Erie, and we have lots and lots and lots of nitrate here too as well. And so we can kind of go down the whole list here of things. And so one of the first things I like to look at when I'm analyzing a genome is really thinking about how it brings nutrients into the cell, right? And because that's going to be the gateway to bring in things like iron, phosphorus, sulfur, carbon, nitrogen. And so I was looking at the transporters of our genome, and a couple things really sort of popped out immediately. We have here ABC style phosphate transporters, and also ABC style transporters for iron and manganese. And so these ABC transporters use ATP to really scavenge elements from the outside of the cell where they're at very low concentrations and bring them inside the cell, right? And so with this, especially here in Lake Superior, having these ABC style transporters is sort of key for this organism to bring these elements into the cell. And once that phosphorus gets into the cell, it actually has genes that allows it to make polyphosphates as well. So this organism doesn't have to use that phosphorus immediately. It can store it as these polyphosphates and break them down whenever they need it for growth. The same thing is likely for iron. A lot of cyanobacteria can form It can store iron intracellularly for long term. And so the gene systems for these aren't as well known, and to my knowledge, and which is very small, I should say. So if anybody out there knows anything about iron storage molecules, please let me know. I'd be very happy to talk about it. But the other things that kind of kept popping out when we started looking at this is the number of transporters for different styles of nitrogen. So we have nitrate, sulfate, nitrite, ammonia, we have urea, we have all of these different styles of amino acid uptake as well with these. And so the amino acids could be used for anything from growth or making metabolites, like say cyanobacteria, things like that. We also have other types of storage compounds for carbon like PHVs or PHAs, and also for nitrogen for cyanophysin. I would also say that anabanopeptin also is a very nitrogen-rich compound that could also be used potentially for storage. However, I think the jury's not out yet on that. But when we looked at this organism under the microscope, we did see potential heterocysts, and we did find a NIF operon within here. And so we know this organism can do nitrogen fixation. Whether or not it was doing nitrogen fixation in the bloom, we don't know. We did not take any samples for nitrogen fixation assays. However, I will say that my student, Keila Natwara, has been measuring nitrogen fixation rates across not only Superior, but the other Great Lakes. And she is finding that we still get nitrogen fixation like year round, essentially, whether it's be late fall, early spring, or deep into summer. Nitrogen fixation seems to be an integral process across the Great Lakes. And so who knows if this organism was doing it, but it did have the machinery in place to be fixing nitrogen as well. And so taken together, I think that this organism is just a survivalist, right? And so it can withstand these different styles of environments, mainly because it has a lot of the machinery that it just allows it to not pay attention to the outside concentrations as much of these different nutrients, and potentially use some of its internal stocks, say when phosphorus becomes limiting and things like that. Okay, so kind of to summarize this, what we just was talking about, I think this organism could be an element scavenging and hoarder. Nitrogen fixation and carbon fixation is just one of the many traits that it possesses. This organism likely is producing anabanopeptin, potentially right before it bloomed or during the bloom. Anabanopeptin was the only toxin that Todd Miller was able to find in that 2018 bloom. And I find it very interesting that this organism can be in very diverse environments, both element ratio wise and also via salinity. I didn't really talk about this, but Finland organisms are commonly found in brackish waters. Okay, all right, so let's shift gears from thinking about the genome to really thinking more about this D. lemmermanii presence in Lake Superior. And again, at the very beginning, I teased that we pulled a 16S rRNA gene from this bloom, right, this Anabana cCAP. And if we did the taxonomy, well, we did we did a phylogenetic reconstruction of this, and we found that it was very closely related to Dolichospermum lemmermanii cultures that were pulled out and 16S sequenced, but not genome sequenced. And again, this whole green box here represents what we would normally cut off as in molecular terms as a species. So using OTU, or operational taxonomic unit, at a 97% similarity cutoff would actually envelop all of these different organisms and genome and 16S genes into one theoretical species. And so my thought was, we've been collecting all this different data, both from sediments and also from the water column in the lab over the years. Let's see if we can mine it using this 16S we pulled from the bloom and see if this organism is present across the lake. And so the data set that we went to was this, our Glenpo phytoplankton sampling. This came from CSMI of 2016. When I first got here, Ewan Reevy and Andy Brandberger brought me on to think more broadly about microorganisms and phytoplankton, and using molecular tools to really understand sort of the distribution across the lakes. And so what we have here is around 30 stations, near shore and offshore. We sampled in April and August. and we do an integrated upper limnion where we take water from 5, 10, 15, and 20 meters, put it all together, give it a good swish, and then we do sequential filtering. Here's our 3 micron filter and a 0.22 micron filter, and we lock those away for sequencing later on. And we've been using the same primer set for all of our data throughout, and so we have data from the 16S rRNA gene using this V4 region primer. And so for a long, and so my graduate student, Jake Callahan, has been working on analyzing these data sets here for a while now, and thinking broadly about the distribution of microorganisms across Lake Superior over time. And so what we were noticing when we were analyzing the data is that we see an increase in diversity and abundance of cyanobacteria in the summer. And this is kind of like a no-brainer. This gets warmer in the summer, right? We know that cyanobacteria and tons of other organisms also respond to temperature. But we did see some of these bigger groups like Aphanizomenon, we have some Dolichospermum, we have some Microcystis, Planktothrix, all starting to show up here in these at some of the stations in the summer months. And if you're interested in thinking broadly about microorganisms at Lake Superior, Jake will be at CHABs next week on Tuesday at 9:15 a.m. In the 2 decades of counting of applying DNA techniques to aquatic sciences, day 2. And so take a look at his talk if you're interested in learning more about microorganisms in Lake Superior. What was interesting about this is that we have these Aphanizomenon and these Dolichospermum and these Planktothrix organisms coming in. These are all organisms in the Nostoc ACA. And so, I was excited about this Dolichospermum coming up, right? Because we're looking for a Dolichospermum. Well, it turns out in the SILA database, the Dolichospermum that we're looking for is not actually classified under Dolichospermum. Rather, is classified under one of these Aphanizomenon. And it's actually this one right here. I believe it's the NIES 81. There's an 81 and a 41. I can't. But I think it's this one right here. This Aphanizomenon. And so when we looked at this clade and looked at the distribution across Lake Superior, I should also back up and we use BLAST approach to really figure out whether or not we had any sequences that were highly similar to our bloom forming sequence. And so when we did this and we blew it apart, turns out all our sequences are falling within this Aphanizomenon clade. And when we looked at this on a map, all of these were across the lake. These aren't actually on land here and here. This is actually just really close to the near shore environment. And because it's a watercolor, kind of bleeds over. But the take home here is that we're finding Dolichospermum livermanii basically across the lake in August. It was at basically every single station. And when we look at this by ASV, so instead of looking at operational taxonomic units that are greater than, lump a whole bunch of sequences that are 97% similar to each other, we looked at ASVs, which are amplicon sequence variants. And so this gives us a little bit better resolution of some of the patterns that could be happening here within the lake. And so one of the big things is that we see both rare variants, so things that are showing up maybe once or just a handful of times, and these are our stations here. So we have our nearshore environments and our offshore environments here. The DCM is the deep chlorophyll max, and this is our integrated water sample here. But you can see across the board, like this ASV 721 is found in every single station we have for in that summertime month, and just a handful of stations in the deep chlorophyll max. So this is really hammering home that we could potentially have multiple versions of this organism floating around the lake at any given point in the summertime. And so this, building on some of that work, this is just hot off the press in the sequencer. Again, we were just looking at spring versus summer months. Last summer, we did this biweekly sampling transect, starting here down by Wisconsin Point, near Duluth and Superior, and worked our way all the way across over to the Apostle Islands and back down over here out here into the bay, or this would be Schwammingon Bay. And so we basically went out and started off in June. In early June, we didn't detect any Dolichospermum limmermanii. And so we're using the same approach where we blasted all the sequences recovered against our Bloom 16S. And here you can see the distribution changes. The size of the bubble represents relative abundance here. And so in late July, you can see things are quite abundant, and then it kind of tapers off again in August and in September, and then we see another sort of bloom here, or increase in sequence, not really a bloom. What's interesting is that here in late July, I was right there a couple days before the sampling of this transect and witnessed a bloom on the beach at a wedding. And so it's kind of like a side note, but it appears that this organism, this Dolichospermum, was present nearly everywhere. And our collaborator Matt Hudson will also be giving a talk on this at JASM next week. I couldn't figure out what this what session was going to be, but take a look for Matt Hudson and the spatial temporal patterns of nearshore cyanobacteria and microbial communities in Western Lake Superior for next week if you're there. And so this is really, really hot off the press. Like, it came off the sequencer just a couple of weeks ago. And so we're still analyzing this to understand more broadly about this organism during the summertime. Okay, so kind of wrapping up this little bit here. I know get moving on time here. I think that's interesting about this and finding these different ASVs is that a lot of these genomes actually carry multiple 16S genes on their genome. For instance, this Philosophiae cCAP has 5 16S rRNA genes. And these things can range in similarity to each other from anywhere from like 98.9% all the way up to 100% similar in these multiple copies. And so when we think about these ASVs, a lot of times they're only 1 or 2 nucleotides off from each other. And so they would all sort of fall into this region. Unfortunately, with metagenomics, it's really tough to say, it's really tough to actually recover 16S genes into a single bin. But it's also tough to say how many 16S copies we actually have on a genome without having a fully complete genome, okay? And so it's really tough for us to say whether, you know, this SB001 has 1 or 5 or 7. But what's interesting though is that we are seeing these differential patterns potentially setting up this idea that there could be ecotypes of D. limmermanii across the lake. So thinking about maybe like an eastern side of the lake versus western side of the lake, or nearshore versus offshore, or maybe something on the watershed. And so this is one of the things that we're starting to think about and really start to investigate with all the data that we're gathering right now. Okay, so this left me with one really big question after looking at all this data and saying, okay, well, it looks like this across the lake, right? It's present down in the lower lakes, Erie, Ontario. You know, is this organism a recent invader? Like, could it have like come across on a boat, be injected out, and then just kind of like slowly taking over? Or could this organism just be a natural part of Lake Superior and it's been here for a very long time? And so I had been doing a lot of sediment work in Lake Superior, thinking about the biogeochemistry of microorganisms here, and thinking how they cycle things like sulfur and nitrogen. And so all along here, we've also been taking and doing 16S sequencing of all the cores here. And so in 2015, when it came to UND, we were very lucky, and we actually got 6 cores from these 6 stations here, all the way from Isle Royal, that's up here, all the way down to right outside the harbor, and right across from the Apostle Islands on the west, and then the eastern side. And so here we can see us on the back deck enjoying some sun. And we also were able to lead date cores from Isle Royal. And so the big question is, I was like, okay, well, we find it in the water column. So using BLAST again, I went through and said, okay, do we find any ASVs present within the sediment? And so lo and behold, we did. The stars just denote ASVs that were found also in the water column. And so you can see we have some that are rare, right, again, and some that are only showing up right now in the sediment data, but not in the water column. But some of these ASVs, like 1797 and also these 3246 and 3266, are quite abundant as we go down core. So when we think about these cores up here at the very top, this is sediment depth. This is the very top of the core, very bottom of the core. We're essentially going back in time, right? So we can start off at present day or near present day, and by the time we get down here, we could be potentially thousands of years old. And so again, we lead dated a core from Isle Royal, which is right up here. And so this is the sort of this brown band represents where we were actually able to get a really good lead to tin reconstruction of time. And so starting off here at about 2015 at the very top, we were able to reconstruct all the way down to about 1880, give or take. And so you can see the distribution of different ASVs here in different colors showing up over time and being deposited in the sediments here. And then breaking this threshold, it's really tough to say how old some of these sediments are, say like from 20 centimeters or 30 centimeters without doing a more thorough reconstruction. But we've done a lot of sedimentation rates across Lake Superior. And so I just took some from Evans et al. In 1981 and just said, okay, they went across all of Lake Superior and generated, I think, around 17 or 18 different profiles. So I took those and I just calculated the median, the mean, the minimum rate, and the maximum rate that we should see for sediments across Lake Superior. And just for as a fun like back of the envelope calculation sort of thing, if we go down here to this one that's close to 30 centimeters, we could be looking anywhere between 300 years and 800 years potentially for when that sequence was deposited or when that aconite or life cell was deposited and sedimented down. If we look at this minimum rate, which happens way offshore out here, we could be looking at something like 3,000 years. I wouldn't say that we actually see 3,000 years. It's probably like a very high end. But I do think thought-wise, it is very provoking and very interesting to think about this organism being present in the lakes, even like say 300 years ago, right? And so, yeah, I think this is sort of like one of these interesting things that allows us to like kind of think back in time, and it's been really fun. And so one thing you might ask is, Cody, are you sure the DNA is being preserved in the sediments? And when I first got here, I had an undergrad that was looking for a project, and I was really curious about this too, because I had just read this paper from John Kirkpatrick in geology, and he was showing that DNA was preserved in these really deep ocean sediments hundreds of thousands of years ago. And these are looking at chloroplast DNA over time. And so I had Austin go out and calculate doing differential style extraction processes. Let's quantify the internal here in brown and then the extracellular DNA. So thinking about a cell that's lysed, it's dead, that DNA gets spilled off into the environment. Thinking about this extracellular fraction as well. And so if we just go down in depth here, you can see that we have a lot of intracellular DNA that quickly goes down with depth. And this extracellular fraction remains somewhat steady as we go down core, right? So we know that cells are getting less and less and less over time abundant. And this is what we actually see in the ocean here as well. And we also sequenced from this fraction. And so we actually have intracellular and extracellular fractions here. And you can see that we're constantly recovering cyanobacteria at very low abundance. And I think this is the key here, is that these are very, very low abundances. They're in the rare fraction. And so if you think about these cells being deposited down and dying and lysing over, you would expect them to be very small proportions of the total community, which is what we're seeing both here and then also in our other data set that we just showed. And so I do think these, in summary, I do think this is a real signature that we're seeing, and really sets us up for thinking more broadly about areas of concern around the Great Lakes, and using these sort of paleo techniques to really start thinking about what did a cyanobacterial community look like 100 years ago, 200 years ago, maybe even like 500 years ago, as we go deeper and deeper in the core. And so some of this is we'll be doing here very soon. And so just kind of check back soon with us, and maybe we'll have another story building here soon. But I think the big take home here is that Dolichospermum has been in Lake Superior for a long time. and it doesn't seem like it's going to be going anywhere anytime soon. Seems to really enjoy living here, and it's just kind of unfortunate that it's blooming. So I think one of these big questions here as we think about this is, what is its ultimate life cycle? And why do we see blooms present here in some years, like in 2018, and not present in other years, right? What are driving these sorts of overall bloom scenarios? And so this is like kind of my future thinking here, is that last year was an interesting year. It was a very low rainfall year for Minnesota and Lake Superior just in general. We had very warm temperatures. And we saw several styles of blooms happening, both within the inner harbor and then also in the open water. So this is the Park Point Beach where I was at the wedding. This is just me taking a picture, and you can actually see this cup. This is where I had my champagne moments before. I scooped up some water just to kind of take a peek at it. And this is what that organism actually looked like under the microscope. But we actually detected inside the harbor, this was a very short-lived bloom that happened within the harbor, detected microcystin and saxitoxin. So, I think the bad news about Lake Superior is that we know the lake is warming. We know chemistry-wise it's changing. It's accumulating nitrate. All these factors are coming into play and saying that the lake is changing, right? And unfortunately, it seems like we may start to see different styles of these blooms. But I would also say, on a good note, is that we have this really great network of researchers in the area that are all keen on understanding these blooms and thinking about the public safety of it. And so by using these sorts of DNA techniques, we're really starting to get a good catalog of who's present in the harbor and also on the nearshore environment and also within the lake, so that we can start making these broader calls about cyanobacterial blooms. Is this a novel bloom from a novel organism? So say something like this Siskawodensis. This is something that we have isolated in the lab, or is it something we've seen before? And it's just been there at very low concentrations or abundances within the water column. So I think moving forward into the future, I think we're really preparing ourselves for being very nimble and being able to react to these blooms and being able to go out and sample them very quickly. So with that, I'd like to thank everybody here for coming and listening to me talk, and also like to thank all of our co-authors on the paper, all of the work that we're doing here on the transect from last year, Matt Hudson, Rhiann, Bob, Sandy, who's been really doing all the sampling and things like that, and our funding from EPA and UMD for this work. So I appreciate you all. Thank you very much, and I'm happy to take any questions you might have.
Okay, Cody, thank you very much for that wonderful seminar. We do have one question so far in the question box, and I encourage others that may have questions to please type them in there and I will moderate them. So the first question we received, John Bratton entered a paper citation. So a recent paper that was published in Frontiers in Ecology and the Environment, and this was by Wilkinson and others. The title was "No Evidence of Widespread Algal Bloom Intensification in Hundreds of Lakes." And John asked if you can comment on this paper, which shows increasing blooms as a result of sampling bias in the study lakes. So it sounds like they found that sampling bias was actually the reason for data showing increased blooms. I'm not sure if I'm reading that into that correctly. And John, I can let you unmute if that's easier as well.
I have not read that paper, and I will definitely go look at it. But I do think sampling bias in all that we do is present. So I think one of the interesting ones comes from the ocean, where it's like, oh, we don't see as much diversity, say, up near the poles as what we do as we do in the tropical waters. Is kind of false, because we only get up there very often, only so often, right? because of winter. And so, you know, I think we have a big bias of just like sampling lakes in the summertime, right? And so we only know about these things that we can actually sample. And so I think you are probably right on, is that some of this is reactionary, like when we see a bloom, right? As we go out and sample it and say, okay, we have a bloom. But thinking about these blooms, it all depends on what is setting that bloom up, right? So how the growing season shapes up, almost like a plant community. You can think that each plant or each species has a different phenology, right? And so what's happening down here, say in may set things up for what's happening in June or July or even August. And so I think sampling bias could be definitely right up there, and something that we should all probably be thinking about. But definitely going to go read that paper now. Thank you for bringing that to my attention.
Yeah, thank you. And I can copy and paste that citation into the chat box here in a second. So our next question comes from Desiree Nowinski, who asks, with the sediment work you are looking at, are you looking at forms and spread of aconites or just Dolichospermum in general? Sorry, I'm butchering names here.
Yeah, so with this, we never had the idea of going out and thinking about aconites being preserved in the sediment, but we are now. So when I first got here, I was really more interested in thinking about the biogeochemical cycling of these organisms, and primarily with sulfur and nitrogen. And so I was really more keen about understanding the organisms that were living in the sediments and doing the cycling, not so much the things that were being preserved. And it was only when we started analyzing these data sets for 16S that I realized, oh, we have lots of 16S. Why don't we go in and try to like pull some of this out from the databases? But I know Bob Sterner's lab is really keen on understanding aconite preservation and whether or not, what is driving these aconites to come back out and bloom. And so thinking about resurrection of these things on a seasonal basis. So yeah, it's definitely something to think about.
Okay, thank you. Next question from Hayden Henderson. Has anyone tried to tie any historical hydrological events to the dating or DNA presence hits from the cores? Curious about your opinion or possible overlap with the frequent flooding and massive external loading events that the western and somewhat central basin of Superior has seen at frequency in the last decade.
That is a very good question and very on point. And so short answer with this work, no. But with what's leading up to this right now, so Bob Sterner's group is looking for these areas that we have somewhat sheltered sediments, right? And so they've been going out and looking for really, really good areas for trying to find these sorts of sediments where you would get good preservation of signal being deposited down. So one of the problems with open water of Lake Superior is that you get these big resuspension events. And so these resuspension events can really redistribute the sediment across very broad areas. And so whenever you see us getting an increase in abundance of that 16S gene down core, we can't really say whether or not it was due to like a big extreme weather event or whether or not it was just because of a resuspension event kind of moving these things around. But I'm hoping that within the next year or so, we'll have a better handle on that and being able to sort of like start to link not just like a 16S gene or an aconite, but also thinking about pigments. And so do we actually see an increase in pigments? Do we see a difference in, let's say like the 13C signature within that sediment? And can we pin that down to an exact age using lead-210 dating and maybe even carbon-14 dating as well? So for this work, we're kind of doing some hand waving, but for this future work, it's really setting that up. So stay tuned.
Okay, thank you. So next question from Janelle Cook is, theoretically, how far back in time could you measure DNA in a core? What conditions would have to be met for good measurements?
That's a very good question in Lake Superior. theoretically, we could go back to the age of when glaciation was happening, right? So that would be our bottom line. Most likely, under a glacier, you're not going to be seeing any of these cyanobacteria, or you would hope you wouldn't be seeing any of these cyanobacteria. And so in Lake Superior, you can actually get down to these, and you can tell where you hit these glacial sediments because they really change in color and texture. And so it just becomes really hard to date these sometimes whenever you get down into that sort of age gap. So we're talking 10,000 years. But at some point, like my colleagues out in the ocean, when they get these really good cores and things like that, like the John Kilpatrick one, 100,000 years, they're finding these sequences that are preserved in that sediment. So, I think we have a big age gap where we can actually start to detect these things in sediments. And so hopefully we could get back to 10,000 years. I think that'd be like the short answer for that question. But it would also be a lot of really good geological work and sedimentation work that would have to happen though to get us back there.
Okay, thanks, Cody. Next question is, how different in nucleotides were most SAVs? Were they mostly 1 or 2 nucleotides or more than that?
Most of them were 1 or 2. I think we saw up to like maybe 3 or 4 was the tops. There was definitely a threshold there. So we basically, when we looked at the separation between all the ASVs, there was definitely a separation where we got down to say the 98% level. Like, so everything was like 98.5 similar to each other ASV wise to that SB001 16S. And then once we got down past 98, it got kind of like dropped down to the lower end of 97% similarity. So probably thinking more about like, we'd have to do the math here really quick, but more like 4 or 5 nucleotide differences within those. And so this might actually be setting up the stage for thinking about different speciation. We know that these theoretical species cutoffs are not always, how do I say this, like, they don't hold for a lot of reasons for many species. And so we know that some organisms can be like 99% similar to each other and still be different species. So thinking about the mycobacterium style groups. And so, it was definitely interesting to see the amount and range of diversity here. But thinking about the genomes, I think that's like for sort of a telling sort of signature is that we do see lots of variation within these sequenced ones that have multiple 16Ss. So when I think about an ASV not being exactly similar, I think there's going to be some float in there depending on the number of copies and how long they diverged and were duplicated on the genome. Hopefully that answered your question.
Okay, thank you. So for our, yes, that was great, was the feedback. Okay, so for our last question, we've just got a couple minutes left, is your final slide gets at a key question about seeing blooms versus not seeing them. It seems like your data suggests Dolichospermum is generally around, but only sometimes forms seeable surface blooms. If so, what do you think is driving the accumulation of these blooms on the surface? Altered hydrodynamics?
Yeah, that's a very good question. And we were just talking about that, looking at the distribution of these and how they change over time in the summer was like really giving us a good inference into like the dynamics of these things. And so I think what we need to do in this next stage is really start thinking about using hydrodynamic models that look at currents and current speed and directions and things like that, and start to incorporate this into our sampling and thinking about where these where Dolichospermum is present. And so you can imagine, like, if we get a shift in currents or get like a deep current maybe pushing these things up really quickly, we might see this expression on shore. There might be some sort of physical trapping where we have interactions with the landscape, right? So we have, maybe that we're seeing them over in the Apostle Islands is because a shift in the currents is really just starting to drive them towards there, and because of the natural geology of the landscape, they're just sort of getting trapped there, right, and getting pushed up into the water, up into the nearshore environment. And so I think there's definitely a lot of questions that we have left to address, and this is like a primary one. So thank you for bringing that up.
Okay, thank you, Cody. That brings us right to 3 o'clock, and want to thank you for spending this time with us today and presenting your research to Sigler and our friends. And for anyone who is interested or has a colleague who may be interested, we will post the recording of the seminar on the Sigler website within a couple of days, so please check back if that's of interest to you, and also encourage you to check out our upcoming seminars. We are holding about one seminar a month, so we hope to see you back here. Have a great day, everybody, and thanks again, Cody.
Thank you all, I appreciate it. Have a good day.
Thanks, Cody.